I can see your screen, not yet full screen, but now it's good. Yes,
Okay, excellent.
go ahead.
Thanks. Well, good afternoon. Yeah, I would just like to start by thanking the organizers and Michael, Alexandra, and Claude for organizing this workshop, and it's been great so far. I really would like to be in Paris. I guess everyone has said the same thing, but hopefully there's going to be more chances, right, in the future. So today I would like just to start by giving you an outline of what we're doing recently. So we're gonna discuss about how we can connect some of previous work in my lab on global sequence models with the idea of the construction of evolutionary models. So how can we kind of extrapolate work done on structural biology and coevolution into new models of sequences? And then, yeah, I'm just gonna give a couple of conclusions on our work. And the first point is that in the last few years, we've been working along with others in the field with the idea of estimating probability distributions of families, right? So if you have a collection of related protein families, it is possible to estimate a probability distribution of the sequences, and this had led to interesting insights on structure prediction, and contact prediction. And this is done by basically assuming that I don't know if you can see my pointer. It's not there, right?
No, actually, yeah, we
Okay.
can't.
Yeah, somehow it's missing. Well, yeah. So the point is that this probability distribution is typically modeled with an exponential that has a set of parameters that include couplings and single side occupancies, and this set of parameters has been useful in answering some questions about the structural biology and function and folding of proteins. Um, Actually, this, what we call this Hamiltonian, can also be used not only to predict structures, but could also be used to have a proxy of evolutionary landscapes, basically understand the variability of proteins and their outcome in terms of fitness. It can also be used for maybe prediction of specificity between complexes, protein RNA interactions, and ultimately also for design. So the point is that this couplings or these interactions that can be inferred from the sequences contain information that captured maybe the evolutionary history of proteins. And one point was to actually try to use those hints, those epistatic contributions, as a kind of driver of a model of sequence evolution. Okay, so that's kind of the premise of the work. But let me first just give you a little bit of overview of some of the origins of the idea of molecular evolution. So in the 60s, Zuckerkandl and Linus Pauling started thinking about this in an objective way, right? So how can we study molecular evolution in some way that is kind of more quantitative and rational, and less based on phenotypical kind of characteristics, but more on a molecular basis. And they came up with this idea that there's a principle, some sort of like a document of history of molecular change that can be maybe kind of taken advantage of to study the evolution of systems, molecules, and so on. And this was basically the premise of the idea of the molecular clock hypothesis. So they said, well, we have several molecules. They propose candidates, and ultimately the ones that prevailed, as you all know, is basically DNA and nucleotides and also polypeptides, because those basically are inherited and they changed, and this change can be tracked, right? So What was very interesting is that even in this paper, which was more like a philosophical paper, they proposed that, yeah, one important idea to deal with molecular evolution is by comparison, right? Comparison of homologous chains. And maybe, the idea that if you have two things that are similar, then we can actually have an ancestor, a common ancestor that can be inferred. So early on, this idea was there. And what I found also very interesting is that at that time they said, okay, in order for this idea to work, we really require to have kind of a large number of homologous sequences. We need to consider the genetic code, which by then it was kind of a new thing. What is the effect of the genetic code in the evolution of sequences? And if possible, also early work was there, study the tree structure with molecular models, right? And all these three considerations are right now kind of extremely important for the study of molecular evolution, and I think that they got it since the beginning. So the point of as you probably know, the idea of the molecular clock can be illustrated in this example, where you have the knowledge of several islands in Hawaii, from geological surveys, when they arise. So what was the time? Like in this case, Hawaii is the oldest, that's 5.1 million years ago. Hawaii is the newest one, have a 0.43 million years ago. And then the idea is you can actually compare genes from fruit flies or birds, and then see what is the correlation between the differences and the time that has emerged. So in this case, it's easier because you know exactly when these islands came about, and you start seeing this linear relationship. So for instance, here near 4 million years of separation, you see that the separation between the genes in Kauai and Oahu, right? That is roughly, that's the difference also in time of emergence, and so on and so forth. So this very nice linear relationship between time and gene separation basically has been used for a long time to kind of fuel this theory of the molecular clock, right? So changes are happening at a kind of pace, and then we can use this as a ticking clock to predict separation of species and time. So it appears to be a constant rate, right? Constant rate of change. But the reality is that actually it is not. So rates really are variable, and we know that sometimes we have bursts in changes depending on neofunctionalization, or let's say if there's a pseudogene, then there's less pressure for it to change, and then you kind of accumulate more changes than other proteins, right? And finally, and interesting to us, is the idea that every protein, every molecule has its own kind of three-dimensional shape, and this shape actually has an influence on and a pressure on the changes that happen, right? So it is not a linear chain, therefore you have very specific constraints that alter these rates. So past that idea of the molecular clock, assuming that, okay, rates are not really constant, then and then with the idea from Kimura that most kind of mutations are neutral to genetic drift, then people started thinking, well, we can model sequence evolution. And one of the first ideas was to say, well, each of the sides are going to change independently, and each of these rates will be independent from the others, and this will lead to kind of a Poissonian distribution of rates. That was one of the first assumptions. However, what happened is that in real data, we really observed that this is not the case. There's no Poissonian distribution of rates. It's more, we observe over dispersion of it. So this was one of the first questions kind of challenging this molecular clock assumption. Then on top of that, then because we just mentioned the idea of the 3D structure of a protein, then people also realize that across sites you also could have different rates, right? and this model called the rates across sides model kind of dealt with that. And the idea is that each of these rates are different but constant, and that led to kind of gamma distribution of the rates. So that was kind of getting a little bit more realistic in the kind of description of what it's observing in reality. But later work by Lopez et al., they basically describe they notice that actually rates not only change across sites, but they also change across taxonomic groups. There's also variability per site. They call this heterotachy. Things are interesting because then you have this kind of set of sources of variability, and people trying to kind of connect these two things. They created a set of models called the covariant models, and the idea here is that you allow a group of sites to change together, and this is kind of fixed, and by design you pick those sites, they change together before being back into something fixed again. And this actually presents very interesting properties, like the rate across sites is not constant, and also you have heterotachy. Finally, more recent work dealt with the idea of you looking at the 3D structure, where rather than setting a mutation rate fixed, you have it as a free parameter that depends on the kind of a stability of the protein. And then you infer the stability with the 3D structure of the protein. Or for instance, you can use a recent model, use more thermodynamical calculations to actually change the probabilities of mutations and based on compensatory changes. So basically a connectivity between the changes. And what they observed was something very interesting, something they call the evolutionary stock shift, where you have some sort of irreversible change after certain mutation happen in terms of the energetics. And these recent approaches require a 3D structure. Okay, so our motivation was, since we think that evolutionary pressures and mutational change are really, really connected to folding and structure. And our point was that probably this idea of inferring couplings, evolutionary couplings, might have a proxy, might be a proxy of maybe the environment or the historic pressures that a family is being exposed to. Then maybe this could lead to some kind of realistic models of evolution. So this direct coupling analysis Hamiltonian maybe can be used to determine these rules of change, right? Given that we know that historically this has served good roles in determining, let's say, structural properties. So our idea was kind of simple. Basically, first get families, infer this global probability distributions through a method like direct coupling analysis, or any other method that actually can infer this epistatic contributions. Then you basically start, you select uniformly a place to mutate, and rather than imposing a probability distribution, you use a conditional probability that is shown here, down here, that actually depends on this on the identity and the couplings of the sequence at that position. So this is going to be changing all the time. It's going to be dynamic. The probabilities will change depending on how the sequence has evolved, and it can be kind of tracked by looking at how this Hamiltonian or kind of fitness, proxy for fitness evolves, right? So here I'm showing evolutionary steps and how the Hamiltonian has been changing based on the evolution. So this is just one example showing how this trajectories actually maintain some energetic stability, but they still keep variability that is important given the changes that happen, right? So the energetic or fitness changes are always there, but in general they're maintained. That's why this is a model of neutral evolution. That's our claim. So here I'm showing you a couple of examples of two families, and this actually were estimated. The couplings were estimated using Boltzmann machine DCA. It's a variant of DCA that uses sampling, and this was developed in Martin Bike's group. What is interesting about that variant is that because it's generative, then the fluctuations are more stable and has better statistics. Although if you use a different type of estimation like mean field DCA, more or less the properties that I'm mentioning now, they're retained. More interesting to us is that this model also captures over-dispersion, just like this structurally constrained model. It actually, the rates are also gamma-like distributed, which is similar to the RAS model. Here I'm showing examples for this response regulator family and the transcriptional regulator LAC-I family. But this actually also works in several other families. Interestingly, we also observe different degrees of heterotachy, just like in these other models. And what is also interesting is the progression of the effective alphabet of these sites. Right, so that they're changing all the time, but sometimes they actually maintain some variability that is kind of connected to heterotachy. And finally, something interesting is that this sequence trajectories also present this effect called the evolutionary Stokes shift. So basically, if we track the changes of a given side, and then we have a way to estimate kind of the energetics of a change. If we let time pass and then we try to come back to its original state, then we observe that we are not back to the original energetic state. So therefore we have this idea of non-reversibility. And this is more evident as more steps happen. And this is observed in different degrees for different families. In this case, the transcription regulator is more evident than for the response regulator family. What is interesting is that this was observed by Pollack et al. And in the context of kind of a thermodynamics model, and it was given by, or proposed to be given by coevolution. So basically these epistatic contributions are the ones that are inducing this effect. And this is also captured by the model. So this, when we look at the, let's say, highest degree of change in the Stokes shift, here I'm showing you some examples of some proteins in these families. We observe that some of these residues with high degree of Stokes shift, they're either close to loops that are functional, they're close to active sites, like the case of the response regulator, or actually, interestingly, they're near transitions of secondary structures. And we believe that the reason is that whenever there's a change in this kind of boundaries, then it could have a big effect on the function of the protein. So if you're modifying this kind of secondary structures, then the reversibility of this change could really be altered. Okay, so and just to kind of summarize, so the idea is that we are kind of introducing a model that uses these couplings as a way to design the mutation selection stage. And instead of like setting this probability as a fixed parameters, most of these properties are actually emerging properties of this conditional probability. These properties include this variability across rates that happens as an emergent property. Over dispersion is observed. Rates are gamma-like distributed. We see the Stokes shift, heterotachy, and all these different pieces from different models somehow get unified in one context. And we thought that was an interesting thing. And interestingly too is that because this is the parameters are coming from a generative model, then these sequences are likely functional, and it's a proxy probably for neutral evolution. So these are sequences that maintain their function, and we believe that there's some interesting applications for this model, maybe in protein design, or you can incorporate it as it's in the inherent model of a phylogenetic tree, or for ancestral reconstruction of sequences. So there are probably more that we're still thinking about. Okay, so I would just like to acknowledge people doing the work, people in my group, especially Alberto de la Paz and Charisse Narti. Alberto is right now living in Paris, so that's a nice coincidence, and the funding agencies. Okay, thank you.
Excellent. Thank you, Faruck, for a great talk. We have a question in the chat from Hector. What is the intuition behind why DCA